Greetings everyone, my name's Steve Hille and I'm here with Fabien Delerue. I lead the FOXG1 Research Foundation in Australia, alongside the FOXG1 Research Foundation in the States and globally. We're here today with Fabien, where he's going to take us back to the basics and run through the genetics of FOXG1 syndrome. He's going to specifically talk about how to read the genetic report, which we all get as parents. And I can speak from experience, it's quite difficult to pick up the report and just start figuring out what it all means, and Fabien will help clear it up. Fabien has just finished running a PhD into the pathomechanisms of FOXG1 syndrome, which was an extremely detailed study, and we're really thankful for him and for Dan for doing that. And we really look forward to it being presented to the public, and we'll make that available. So I'll hand over to Fabien and he can kick off the presentation. Thank you, Fabien.
Steve, so hopefully soon we're about to publish that, and we're really excited about it. But really today, I think the idea is to help the families first and foremost to, as you mentioned, read and understand better their report. When families approach me, most of the time, the first question they ask was about trying to actually understand the report, which is the first thing they are provided with. And it is indeed quite complex because the language that is used on the report is the language that geneticists would use, obviously not families, they're not used to that. So the idea here today is to provide the basics of genetics that allow the families, hopefully, to better understand and kind of decode the report. So to start with, when the families receive a report, it looks something like this. And you see a bunch of letters and numbers here. And it doesn't immediately make sense to those families. So we'll try to take it step by step and analyze the important information that is provided here on the report. But for that, I need to provide you with a bit of knowledge of the genetics underlying this report. I will try to do it as simple as possible, but as you can imagine, the genetics is quite complex. So hopefully it will be accessible and understandable to everyone, really. That's at least the goal. So talking about simplifying, if you take a cell, and you see here on the right an animal cell, and the kind of cells you have in your body, if you want to reduce it to the strict minimum, the cell would be, as you see here in blue, let's pretend for now that this is just water. We actually call that the cytoplasm, because this is not water only, but let's simplify that. And the second important compartment of the cell inside the cell is here in purple, and this is the nucleus into which the genome is contained. So the genome is located inside the nucleus. This is the same for plant cells, by the way. Now, if you talk about genetics, you will essentially talk about genome and DNA. And once again, to cut corners, let's for now assume that a genome and the DNA is the same thing. These are synonymous for now. And what the DNA or the genome is made of, it's actually a long stretch of 4 different elements that we call nucleotides or bases. Once again, let's assume for tonight that those 2 names are identical and synonymous. So, and these 4 different bases or 4 different nucleotides are the famous letters A, T, C, or G. So the DNA or the genome is a really, really long stretch of these 4 letters. And it is represented here. It is, by the way, a Nobel Prize from the 1950s. So what you see here on the right is the so-called double helix that has been discovered by Crick and Watson, for which they had the Nobel Prize. But really what the DNA is represented on the left here is this really long chain of the 4 different letters. And I just want to, one of the main difficulty in biology is to kind of understand at what scale we're talking about, because there's the very, if you start from the very big and going to the infinitely small, then you have different elements. And to give you an idea here, in your genome, in every single cell of your body, so within the nucleus, the DNA is made of 6 billions of these nucleotides, of these letters. And this is contained in a nucleus that is only...
Sorry, Fabien, I think we just lost you there for a sec.
Sorry, which...
Yeah, I think we just lost you there just momentarily. You were saying there's 6 billion letters, is that correct?
Exactly. And what I was saying is that in this, these 6 million billions, so billions is, by the way, 1,000 million nucleotides, so quite a big number. But surprisingly, these are contained in the nucleus, and the nucleus is only 6 microns, which means 6 thousandths of a meter. So it's something really, really tiny. And if you were to actually free this long stretch of DNA within one cell, pretty much you would end up with 2 meters of this long stretch. And if that is for one of your cells of your body. But if you take all the cells of your body together and you put the DNA all together, you would end up with twice the diameter of the solar system. So the reason for me to say that is because you already grasp the complexity of, if you have a genetic disorder, of understanding at what scale we try to understand the problem, and also, yeah, really the complexity of trying to decipher and understand all this. So there's a lot of things that we don't know yet about genetics, and it's based on the technology advancement. But we still know quite a lot, and we know enough to provide families with a report. And so to go back to my first explanation, here you have a schematic of this genome, so this long stretch of DNA. And as you can see here on the right, you have these 4 different bases. And this piece of DNA is sometimes assembled into what we call chromosomes, which are segments of this long stretch of DNA of your genome. And this is stored, as I said before, inside the nucleus of a cell. But now that I kind of described what the genome or the DNA is, I want to focus now on this, which is the concept of a gene. And the gene, you will hear that all the time, is simply a really small and tiny part of the entire long stretch of DNA. Genes are portion of the DNA that encodes the protein, and this is why they are so critical and so important. However, despite the fact that there are 20,000 genes in the human genome, so one can imagine the complexity of what we're talking about, it is, however, only 2% of the entire DNA, of the entire genome. But these small segments, these genes, are really critical because the protein are pretty much the block, the building block of the cells, and that is what defines life. You would not find protein in anything that makes up the world, which is not alive. So if you look into the chemicals of a rock, a stone, there is no protein in there. But what makes you different than the rock, what makes you alive, is actually the protein. So that's why genes, which encodes for the protein, are so important.
I mean, just to bring it to like our situation, I suppose, FOXG1, my understanding is that the reason this is really important is because the instructions to create the proteins are a bit off or have some issues because of the specific mutation, via the FOXG1 gene. And my understanding is that because of that mutation, the instructions are wrong and the proteins either don't, there's some issues with those proteins. Is that right?
This is exactly the purpose of my next slides. You got it absolutely correct. And the idea is now to provide you with how the cell goes from the gene, so the different letters, to producing a specific protein. And the protein, unfortunately, if you change the DNA, the genome, this will have the consequences of changing the protein ultimately, because there is a code that in between those two. So obviously, if you change one, you will change the other. And the consequences can be sometimes what we call silent. A lot of mutation actually do not affect individuals in terms of developing a disease. And actually, the different mutations are somehow required for life. That's what makes us all different. But unfortunately, some of the mutations within the genes particularly are those who induce, unfortunately, disorders, diseases. But let me go through the actual code and the relationship between the DNA and the protein. So here, to simplify, once again, at the top, you have the DNA or your genome. You would imagine that this is actually, once again, the entire sequence of A, T, C, G, etc. And ultimately, here there is the protein. And as I just mentioned before, these proteins are so critical because they are the blocks, the biological blocks of life, that they have a function in the cell, or if they don't have a function, they are part of the structure of the cells. You have the small membrane that I showed at the beginning in the first slide, are made of protein, essentially. So the idea really here now is to try and understand the relationship between the genes and the protein. And as you rightly mentioned, if there is a problem in the gene, then ultimately you also have a problem at the protein level, and this is what creates the disease. So now let's go back so that you understand. Let's go back to the very structure of the gene. What exactly is a gene? The gene, I put it here on this small diagram, on this small schematic. When I'm asked what exactly a gene is, at least the structure, I like to take an analogy. And I usually take an analogy and compare a gene with the train, because the gene is made of different elements. The first one here on the left is called the promoter, which if you take the analogy of a train, would be the locomotive here. Then you got what we call the exons, these blue boxes here, which would be the equivalent of the cars or wagons, I think you call them. And in between the introns here would be the equivalent of the link between the wagons. And why am I now specifying the structure of a gene? This is because it is why we discriminate all these different parts within the gene is because when you go from the genome, so at the top, the gene here, to the protein, the process within the cell is done in two steps, two very critical ones. The first one is what we call transcription here, and the second one is called the translation. There is an intermediate between the DNA. So this, just to remind you, we're still talking about a stretch of these 4 letters. So there is an intermediate between this and the protein that it encodes, and this is what's called the RNA here. You've heard that because of the COVID vaccine, which is based on the RNA. So everyone's talking about this at the moment. The only difference between the DNA and the RNA, and there are actually 2 differences, is that, as you've seen before, the DNA is a double helix, and it's made of 4 different letters. The RNA is actually a single strand, and it's made of almost the same letters. There's only 1 letter that is different. But the very important phenomenon that happens once the DNA is encoded into RNA, before it becomes a protein, there's a phenomenon here that we call the splicing. And simply by looking at this drawing, you would understand that the introns here, the red parts, are just simply cut out, excised, spliced out, we say. And this is why, ultimately here, you end up with a sequence So we're still talking about these 4 different letters. That is different than the original one because you have spliced out, excised, deleted some letters here, which made up the introns.
So Fabien, just quickly, the introns, do they carry instructions that aren't needed when you get down to the splicing, or they never carry instructions? What are they, just connective sort of?
So that's so when at the very beginning I was mentioning that the genes are made up only 2% of the genome, but they are the most critical because they encode proteins. This is a dogma for the last 50 years plus, but this starts changing because we realized over the years that the non-coding part, which the introns are part of, right? These introns are removed. And for 50 years, we thought these parts are junk. They're completely useless. They have no function. But this is not true. I have another program which looks at the role of long non-coding RNA. So typically it's made of these red parts here, but much longer. And they play a major role in life and sometimes in disorders, in diseases. But for the sake of for FOXG1, it is not that the introns are not that critical. Let's put it that way for now. So let's assume that we can discard them and they have no role whatsoever. So indeed they are discarded through splicing, and then eventually the remaining sequence here will be translated into protein. This is once again represented here, and as I mentioned before, the only difference between the DNA is that it is made of a double helix, whereas the RNA is a single-stranded, and also, as I just mentioned, the DNA is made of A, C, T, and G, whereas in the RNA, the T is replaced with a U. All this doesn't really matter, but I just wanted to mention it for the sake of that presentation. Now, let's once again try and put that into perspective. If you go back to at the cell level, at the cellular level, all this, the first step here, which is going from the genome to the RNA, happens in the nucleus. However, the RNA goes out, and the translation from the RNA to the protein here happens in the cytoplasm. So you can see immediately that when I told you at the beginning it's just water, it's actually not water. It's a very complex mix of very important elements that allows the translation of a code here that is contained within the nucleus into functional proteins that are into the cytoplasm. And as described here, you already see that a protein is actually made of, you see all these different modules which are called amino acids. And there is a relationship between the actual sequence of the RNA and the corresponding amino acids. So if you, as you can imagine, if you change any of this letter, this will change the amino acid, and consequently this will have this will change also the protein, and that's when problems can occur. So I bring all this information, hopefully it's not too confusing, to really show you that there is a code, and the code has been worked out over the years between the genome, the DNA here, and the final protein. And the code is based on what the triplets, so 3 different bases, and those 3 different letters code for 1 amino acid, which make up the proteins. So the code is given to everyone freely now, and you can see it here. If, for instance, the letters would be UCU at the RNA level, then that would code for 1 amino acid, which is called a serine, right? However, if you, instead of reading UCU, the next 3 letters are CAU, then the next amino acid that will be incorporated into the protein is an histidine. But surprisingly, every single protein on the planet, and no one really knows why, always start with this specific triplet here, this codon, which is AUG, and they always code for a methionine. No one knows why. This is by itself a subject of research, which is really interesting. And this is all across all the living world. But you would also notice here that, for instance, so proteins always start with this triplet, but they stop with three. There are three different triplets or codon that indicates that this is the end of the protein. And what it tells you is that there is redundancy. So for instance, here, UCU code for serine, as I said, but UCC or UCA also code for a serine. So that tells you that some mutations will be what we call silent, because here, if you replace the U with a C, you still end up with the same amino acid and therefore the same protein. Whereas if you change UCU for UAU, then you change a serine with a tyrosine, and that usually has drastic consequences. But really, I wanted to bring you to this because this is very important, because this is the key that allows you to go from the genome, the DNA, to the protein. And that, let's go back to the very purpose of this presentation, allows the family to understand their genetic report. Before I go back to the actual report, I just want to show you the sort of mutations, because we're talking about changing a single letter that can have important changes on the protein. If you imagine that these letters in the genome are like the letters of the alphabet, and you see a sentence like this one here, as you can imagine, changing a single letter sometimes change entirely the meaning of the sentence. And that is what happens as well at the protein level within the cell. So on the FOXG1 Research Foundation, this screenshot here comes from the FRF website. You see that there are different types of mutations. And for instance, you'll come across what we call missense mutation that sometimes is indicated on your report. And what it means in that case is that a letter has been replaced by another, and that changes, as I explained before, the amino acid. And that usually has a massive impact on the protein, and therefore can cause disease. Another type here, as I explained before, is if, unfortunately, the mutation, the change in this letter induce a stop, then instead of having this long protein here, which is the normal one, the protein is what we call truncated. So it stopped earlier than it should have. It's a different type of mutation, but it still has massive consequences most of the time. And finally, what is probably the easiest to understand for families is if you have insertions or deletions, so you add or you remove a letter as it is here, this can once again have massive consequences and change the protein. In that case, we're talking about FOXG1 protein, and then unfortunately have consequences.